I'm an adult hematologist, and one of the areas of unmet need in our specialty is relapsed and refractory acute lymphoblastic leukemia. Essentially, whenever patients relapse after having had a bone marrow transplant, there's very few treatment options available to them. And the reason we were looking at CD19 CAR is because, of course, there's been huge success with CD19 CAR T cells for pediatric and young adult ALL. So we thought that it would be a good population to look at for this type of therapy. Now, the problem with adults who have relapsed and refractory ALL is often they're older and they have quite a lot of comorbidities, and they can be particularly vulnerable to the side effects of treatment. And we all know that CAR T cell therapy can be quite challenging to withstand. There are toxicities associated with this treatment. So what we've done at UCL is we've designed a type of CAR T cell that has a fast off rate from CD19. And by that I mean that it has a different binding kinetic to some of the other conventionally available CARs. And in having that sort of rapid binding kinetic and the fast off rate, it means that essentially it behaves in a more physiologic manner. So it means that it doesn't necessarily secrete quite as many cytokines as the other available CARs. And this, we believe, has a sort of a knock-on effect on the risks that the patients will have of cytokine release syndrome from this CAR T cell. And because actually cytokine release syndrome is probably one of the biggest problems in ALL. Actually, it's one of the most challenging things to manage. So that was the kind of the ethos behind why we felt a novel CAR was indicated, a sort of a low tox CAR, if you like. And it was with that we sort of developed the study. What we did was, we've got a 20-patient study. It's a phase 1 study. And the patients are enrolled in, and of course it's an autologous CAR T, second generation 4-1BB zeta CAR. And the manufacturing takes approximately 4 weeks. So the patient is leukapheresed, and then they undergo bridging therapy. And then once the product is QP released and it meets criteria, the patient is admitted to UCLH for treatment. And effectively what we do is we give a period of lymphodepletion treatment. So that's chemotherapy to effectively sort of create a space, if you like, for the incoming CAR T. And that's fludarabine and cyclophosphamide. And then we designed the study in such a way that safety is really at the heart of it. So the first thing that we do is we look at the burden of ALL that the patient has before they come to us. If they have high burden disease, and by that we've defined that as sort of more than 20% of the leukemia blasts in the marrow, then we give a lower initial dose of CAR T cells. The second thing that we've done is that we've split the dosing so that it's just a sort of like a safety design, so that if the patient gets any sort of toxicity over the first 9 days following the first dose, that we have the option to withhold that second dose and not contribute to any further toxicity. So we felt that design was kind of protecting this at-risk patient population. So those are the two safety design features. And then the patient, essentially, we evaluate their disease burden at month 1. So we look and see what the CAR T cell's been able to do at that point. And then the follow-up's ongoing then for the next 2 years on the study. So, so the kind of patients that we recruited into the study, again, they were sort of patients with adult B-ALL. And the median age of our patients was about 43 years. And they were a population of patients pretty heavily pre-treated with chemotherapy and transplantation before. So 47% of our patients had actually had inotuzumab before, and 26% had been exposed to blinatumomab, and 63% had had a prior transplant. So, you know, these patients have been through a lot of treatment. And actually, they also had quite a heavy burden of disease when they came to us. So 42% of them had more than 50% blasts, and 21% had between 5 and 49 blasts. And the more sort of disease you have, the more high risk you are of toxicity from treatment. So we were aware that we were going into this with quite a sort of a high risk profile patient population. And in terms of the manufacturing, so again, this is a phase 1 study. So we actually, it's not a commercial product. So we manufactured this in-house, and we used 2 different types of manufacturing platform. We had an open process, which we found to be quite labor intensive, and it was very difficult to scale to the number of patients we wanted to treat. So through the study, we switched over to a semi-automated closed manufacture process using it's a platform called the Miltenyi Prodigy. And we found that to be hugely helpful to us because we were able to turn around patient products much more quickly. And we found that actually the characteristics of the products, they were very heavily skewed towards memory T-cell populations. And actually that seems to be pretty important for CAR T-cell therapy. You want to sort of have a good memory population to enable your CAR to persist in the patient. And that is important for B-ALL, where persistence is really pretty key. So that was the manufacturing. And then the next thing that we checked really on the study was whether or not the CAR T cells did persist. And we were able to do that just looking at the DNA for the CAR in the patient blood. And the nice thing about this was whenever we got patients over the 12-month mark, we saw that actually the CAR was still present in the blood using this DNA analysis. And we were also able to look at the pharmacokinetics of the expansion of the CAR T in the blood as well. And we noted that it was really, it was very comparable to that was reported on the Eliana study. That's the sort of pediatric and teenage young adult pivotal trial of CD19 CAR for B-ALL. So we're very happy with that, indicating that our CAR can expand equivalently to, and perhaps even slightly more than what's perceived to be the gold standard CAR. And not only that, it persists in the blood. But I think probably the most important thing really is the fact that, when we use this CAR, the safety profile was really excellent for this. I mean, we've already defined the patient population as being high risk for toxicity by virtue of the amount of disease that they have in their bone marrow. And actually for cytokine release syndrome, we didn't actually see any grade 3 CRS on our study. And that compares really favorably with what's out there from other CD19 CARs for ALL. The fact that we didn't see any cases is really quite, we were very pleased with that. We did see grade 2 CRS. We saw that in 7 of the patients, and that usually defines patients who've got a high fever and maybe they've had a drop in BP and their blood pressure associated with that. But that was really the worst extent of CRS that we saw. And we did use tocilizumab in a proportion of our patients, and 32% to mitigate CRS. And those patients that did get the grade 2 CRS, they all had heavy burden disease. Very easy to sort of predict who may potentially develop that complication from the outset based on the blast percentage. So that was that side of the toxicity. And then in terms of neurotoxicity, that's another big problem with CAR T cell therapy, and particularly neurotoxicity that's very slow to recover with appropriate therapy. And so what we actually saw, we saw this sort of neurotoxicity arise in 4 out of the 19 treated patients. And we saw 1 patient with a grade 2 event and 3 patients with a grade 3 event. But I think, again, the important thing to note is that the neurotoxicity responded really swiftly to the introduction of steroid treatment. And so we had a couple of those patients who had the grade 3 events. They resolved to grade 1 in less than 24 hours with steroids, and the other one within 72 hours. So again, that sort of, it was quite easy to manage that syndrome in some ways. And it was also very easy, again, to predict who was going to get it, because it was the patients who had the high burden disease who went on to get the neurotoxicity. And all of those cases indeed were preceded by CRS as well. So you've got sort of 2 pieces of information that help guide, and you can be, I guess you can be more vigilant with those patients, knowing that they're potentially at higher risk downstream. So that was the kind of incidence of the common toxicities that we see with CAR T. There were cytopenias, as you see with all CAR Ts, but most of them did respond by about month 3 and had normalized. So we were very heartened by that tox profile on study. And what we did see was that in our current cohort, 11 out of our 19 patients, that's 58%, are currently disease-free, and that's at a median of 12.2 months of follow-up. And I think we just have to kind of put this in the context of this is a patient population for whom there really are no other effective therapies. And So I think this really does, you know, it's really quite an important piece of work. And I think the other thing to say about this is the fact that these patients are in remission, but without having had a sort of a further line of consolidation. So some people argue that maybe a bone marrow transplant or a second bone marrow transplant might be indicated if you do manage to achieve a remission with a CAR T. But in our hands to date, you know, there's been very few of these patients have actually had a bone marrow transplant, and we've got 5 or 6 of them who are over the 12-month mark without having had another line of treatment. So, you know, watch this space. I guess we're sort of, you know, watching the kind of the patient's progress on this, but we're very encouraged to date by what we see. So, you know, I guess what we've learned from this study to date is the fact that, you know, the manufacture method, the in-house sort of semi-automated manufacture method seems to be pretty good. It makes a good product. It's got lots of memory, T cells, and it seems to expand really nicely in the patients. And we saw really nice persistence of the CAR Ts, which we think is probably helping to protect patients against the risk of relapse downstream. And really of note the fact that, you know, it was very tolerable from a sort of a safety perspective. So patients weren't getting, you know, very severe cases of CRS and neurotoxicity. And in fact, where neurotoxicity arose, it responded really quickly to steroid treatment. And that's, you know, very reassuring for us. And from the point of view of efficacy, you know, 84% of our patients achieved a complete response to treatment at 1 month. And it just appears to be durable, you know, as I say, 58% sort of disease-free at a median of 12 months. So with this information, I mean, we hope to invoke a global phase 2 study. We're sort of looking at taking this to a bigger population of patients. So the manufacturing will no longer be done by my small team at UCL, but will be done on a bigger scale. And hopefully we'll just get to confirm these findings and perhaps be able to offer something to adult patients with ALL who really, you know, don't have many other treatment options available. Well, we're also looking at CD1922. I mean, we're, you know, because the thing with ALL, so one thing that people always want is to optimize the CAR T cell product itself, and we're looking at lots of sort of, you know, different manufacturing technologies and additives to be able to make the CAR expand even more and to essentially enable even greater persistence. And we're aware of the fact that on this study, and like any study targeting a single antigen, that antigen escape is one of the Achilles heels of the treatment. And so there have been 3 instances of CD19 negative relapse in this study. So my next plan is to try to create a CD1922 study to see whether or not we can try to prevent those CD19 negative relapses from happening. So we've got, you know, manufacturing, I guess, progression, and we've also got this sort of, you know, antigen targeting approach to try to minimize CD19 negative. That's our big next agenda points.